No. So there is a phenomenon called cytoplasmic polyadenylation, but that is not the norm. In general, what you have is the poly-A tail is created in the nucleus. It is just the poly-A binding protein changes. There is a nuclear version of it and a cytosolic version of it.
Okay.
Does that answer your question, Kunal?
Sir, actually, my internet speed is down, sir. Actually, the sound is breaking.
Could others hear it?
Yes, sir.
Okay.
Okay, sir. I'll
So then you take it from the recording later on. Today, internet is giving trouble. Even my speed is not good.
sir.
Sir, and in I think lecture, I don't like the first set of lecture in post-transcriptional modification.
Mhm.
You have talked about the housekeeping genes. So because of the poly and the capping and the like tight regulation, the half-life or the stability is more in housekeeping genes. But you also
Mhm.
talked about cytokines,
Uh-huh.
their mRNAs are like less stable.
Yeah.
How, sir? Like their poly A tail is small. That's why.
So it is, I don't think it is entirely clear, but yes, their shortening of the poly-A tail is faster. That is one problem. And in certain cases, they do have the stop codon, the natural stop codon in the last part one exon.
Sir, the last part, can you come once again? The last part I
Huh?
did not Sir, the last line we told, I did not understand that line.
That some of them
Hmm.
don't have the stop codon in the last exon, but in an exon before that.
Okay, okay, okay. Okay, sir.
But I don't
But
think the
it's not.
detail of the regulation is known.
Okay, sir.
That how their half-life is short.
Hmm. Okay, sir, understood.
Hello, sir.
Uh,
Sir, like I'm little confused about something. In the lecture you mentioned that, I mean, somewhere you mentioned that the transcription happens up to poly A tail or something like that,
no,
and after that the region, the stop
no, no,
codon.
no, 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 no. Transcription happens up to the termination site. Then it is cleaved at the polyadenylation site because uh, the cleavage factors are recruited on either side of the polyadenylation sequence.
Sir,
Okay.
so we talk about like reading frame and all those things. So the sequences which are beyond the poly A site are not required for the protein production.
Of course not.
Okay.
In fact, even before the polyadenylation sequence, like signal,
Hmm.
those are not part of reading frame, like coding sequence.
Okay.
So what is the mechanism of stopping transcription uh, at stop codon? Like,
By
and how does that
there
remove?
is no stop codon for transcription.
Yeah, there is no stop codon for transcription.
It is a poly-U rich region. Remember when we were talking about mouse genetics for conditional, you had this stop cassette between the constitutive enhancer and the reporter gene.
Yeah, yes, sir.
The same thing.
Okay.
Looks like most of you are seeing the slides for the first time now.
Sir, and one more thing, like you said, like there are always RNA binding proteins. I just
Yeah.
want to ask, sir, it may be a silly question, why free RNA cannot exist in the cell? Why it has always to be bound by proteins?
Because RNA has that 2' hydroxyl group as well.
Yeah.
So if you give it access to hydrogen bond donors, it
Hmm.
might undergo hydrolysis very fast.
Okay. So that is why it is in a way protected by the
Yeah,
binding proteins, RNA
yeah.
binding proteins. Okay, sir.
Sir, you also said that mRNA usually remains in the cyclic form. So after
Non-covalent cyclic form.
yes, sir,
Non-covalent cyclic
non-covalent
form.
cyclic form. Sir,
Hmm.
sir, it remains just after transportation into cytoplasm, or first translation cycle happens and then this process happens.
Yeah, so the existing data suggests that it enters the cytosol as in cyclic form.
Okay.
Okay, sir.
Yeah.
Sir, and
So
one more
then
thing.
at the time of translation, it opens.
Mhm. It remains in the cyclic form. So there is also speculation that this non-covalent cyclization improves the efficiency of reinitiation, meaning the ribosomes that were translating, they will fall off at the stop codon, right?
Yes, sir.
Now, if it is cyclized, then the stop codon is very close to the cap site. So it is captured again and used for the next round of initiation.
Okay, sir.
Means same RNA will be used to produce multiple
Protein
rounds of
multiple times.
proteins. Rounds of
Yeah,
protein.
when I will teach you translation,
Hmm.
I will introduce you to a term called polyribosome or
Hmm.
polysome
Hmm.
that talks about precisely this.
Okay, sir. Sir, and in the capping section, like we have talked about the 7-methyl guanosine cap. After that, also there is a step of 2-O-methyl transferase, which adds the like methyl group to the last
The
what
first,
is
the
yes,
first nucleotide
nucleotide. Yes.
that was transcribed. Yeah,
Hmm. So why that again is required? You
I do
already
not.
have.
Yeah, I do not really know that.
Okay, sir.
Hmm.
Hmm.
In the 5' capping, how does the capping happen between the 5' phosphate?
Enzyme है करता है.
So but there should be some chemical reaction happening.
It's an esterification reaction. It's an it's a sorry. It's an anhydride reaction.
Okay,
Phosphoanhydride bond is formed.
okay, yeah.
पढ़ाई नहीं किया हो इसलिए उल्टा सीधा सवाल पूछ रहे हो.
Sir,
You're
and
not.
one more
None of
thing.
you are asking in-depth questions. You're asking extremely superficial questions.
Sir, and one more thing. You said one line in the lecture that RNA DNA duplex are more stable in the DNA DNA duplex. Why,
Yeah.
sir?
Well, I do not know why, but let me check that whether it is correct. Yeah, so there are a whole lot of papers on it. but I would not know the technical reason.
Okay, sir.
Okay, do you see this?
Yes, sir.
Yes, sir.
There are more. So, like if you search the net, you will get many such papers. And these are fairly old papers. And that is the basis of, like that R loop formation.
Yeah, yeah, sir.
It's of course an artificial system, you understand that, right?
Hmm.
Sir, sir, can you explain this sex determination in Drosophila one more time? Sir, actually, in one slide, I was not able to understand some parts.
Yeah. Okay, so you can see it, right?
Yes, sir.
Yes, sir.
Let us start from here. That eventually what you need is this protein, double sex. Double sex, either it should be the female version of the double sex or the male version of the double sex. Now, to get this, here is the issue that whether exon 4 will be included or not. If exon 4 is included, then it has its own polyadenylation sequence.
Sir, here I want to ask, sir, it has its own polyadenylation sequence. What that does, means it is already having a like termination sequence or something like that.
Yes, it already has that AUA and all
And
the
the GU and yes, the whole stretch it has.
yeah,
Okay.
yeah,
Okay.
yeah. So if exon 4 is included, then the protein ends right there. That's the female version. If exon 4 is not included, then you get the male version. Now, for exon 4 to be included, you need the SR proteins, okay, to be recruited there. Now, these are SR proteins, RBP1 and TRA2. But RBP1 and TRA2 cannot bind the EACs of exon 4 until and unless TRA is bound there first. Now, TRA can only be produced in the females. Why? Because exon 2 of TRA has a premature stop codon, in-frame stop codon. So if exon 2 is included, then full-length TRA cannot be made, which happens in males. But in females, because exon 2 is not included, and in this case, there is a splicing inhibitor. So the female specific, only in females TRA is made. This splicing inhibitor is nothing but your sex lethal itself. Sex lethal is a splicing factor. So if sex lethal is there, it prevents the incorporation of exon 3 of its own gene or exon 2 of TRA. Now, whether the sex lethal will be produced or not is dependent on the ratio of X chromosome to A, the autosome. The X chromosome produces the activators, the cis A, B, etc. And autosome produces the inhibitors, deadpan, etc. So if it is two X's, then some of the numerator factors will be engaged in a dead-end heterodimer with deadpan, but some will still be available to recognize the early promoter of sex lethal. But if there is only one X, then all the numerator proteins are engaged in a dead-end complex, so no transcription happens from the early promoter. Now, only if and only if the transcription happens from the early promoter, then this sex lethal is produced, which I also do not know very well, is that how is this excluded? Like when sex lethal is not there, how is this excluded from the early transcript, mature transcript? I also do not know that.
So
But
because they are also that XL protein will not contain the male specific exon, है ना?
yeah,
It should
but
not contain.
if you think about it, it is before the first molecule of sex lethal is produced, right?
Yeah.
Until and unless we assume that there is some sex lethal coming from before. I do not know the exact detail of that.
So,
But
here one more point I want to ask, like what is the differential like regulation of a early promoter or late promoter? And like early promoter, why it is present downstream of the late promoter?
see, that by itself is not a big deal. It can be present downstream or upstream. That is not a defining factor.
Okay.
But what is a defining factor is that the AUG is there only after the early promoter. So no matter what you do, your protein will start only from that AUG. But the fact is that from the early promoter transcription, the sex lethal that is produced, that is there only in the females, and that allows the exclusion of the third exon. And as a result, you get the sex lethal protein in the females, but not in the male. So that's what is the bottom line. But this part is a little unclear to me, that why in the transcription from the early promoter, the first rounds of transcription, why the second exon is excluded there as well. What is preventing its inclusion there? I really do not know.
Okay, sir.
Sir, and here the RBP one we talk about is not our RNA polymerase 2 RBP protein.
No.
It is a different protein. No? Okay.
Sir, 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 these early promoter and late promoter region, are these two regions are separated in two loci or at the same loci?
Same loci, just separated by few kilobases.
Okay, sir.
Sir, and one more thing, like we are talking about poly A-tail. Just this question came to my mind: why only a poly A-tail? Why not a poly G-tail or a poly C-tail can also stabilize the mRNA?
As I said repeatedly, that there is no God that sits in a place and designs how the system should work. Yes. This is a result of millions of years of experimentation, right?
Yes.
So at some point, poly A-tail was added in an experiment, and there must be a co-evolution of a poly A-tail binding protein. Now this combo was doing the job, so there was no evolutionary pressure to come up with another mechanism. That is the only explanation that I can think of.
Yes, sir.
So nowadays people are doing synthetic biology.
Yes.
So if you can make a protein that will be binding to poly G
Hmm.
and will have the ability to interact with cap, like cap bound proteins. It may very well do the job.
Yeah.
But the whole machinery has to be evolved all over again.
Sir, and one more thing, can you explain that part, like phosphorylated CTD helps in the recruitment of the spliceosome components, and then these proteins, the HNRNPs, enhance the interaction of RN polymerase 2 with the elongation factors.
Yeah.
Yeah, so that part, sir, can you explain a bit?
इसमें करने का क्या है इसमें? That
Hmm.
those splicing factors, they have specific affinity towards elongation factors. Khatam.
Okay. And during that, the serine 2 is phosphorylated, na sir, in the CTD.
Yeah. Serine 2 and 5 both.
5.
Perhaps.
Okay. Yes, sir.
Sir,
These are
does
not-
poly A form? Yes.
Huh?
All right.
Sir, does poly A form any secondary structure like G quadruplex or something?
No, not that I know.
So poly C also forms secondary structure. So can that be a reason why poly A was selected?
Just poly C, does it form secondary structure?
Yes, sir. I motif it forms, right?
I have no clue. See, like there are only a very limited set of things that I know, and majority of the things I don't know. And I'm very happy with that state of mind. So, yeah, poly C, what kind of secondary structure it forms?
It forms I motif.
I want it.
Let me check. So it says that if there is a poly I stretch, then only it forms a secondary structure. Just a poly C stretch by itself, are you sure it forms a secondary structure?
Yes, sir. Actually, during my master's, we had a chapter on the G quadruplex and I motifs. So that's how I came to know.
Okay, please share something with me so that I can take a quick look at it. Not that I want to be a master of RNA secondary structure, but it is interesting information. I just want to understand the science behind it.
Yes.
And sir, one slide talked about that splicing is carried out by ribozymes. So that is in case of group 1 introns or group 2 introns?
Ribozymes.
Yes, sir.
Ribozymes will be your group 2 introns, like where you actually have separate snRNAs, etc.
Okay. Okay, because they are like the snRNAs are only helping in splicing, so they are termed as ribozymes.
They are talked as ribozymes, yeah.
Okay, and group 1 introns have the self.
I think perhaps both can be called as ribozymes.
Ribozymes, because
Yeah,
there
because,
also it is the
yeah,
self.
because see,
Yeah,
the ribozyme, matlab, the RNA is acting as an enzyme, which
enzyme.
is true
Yes.
for both.
Yeah. Okay, sir.
Anything else?
So, like in general, when there is poly A tail, after a loop formation, like in which that in the end of transcription, right? Like the whole poly, the loop is able to the loop gets heavy or something like that, and the poly tail is removed.
What?
So, so in the
Prokaryote and eukaryote?
so there is a loop kind of thing before the termination of, so how the in the machinery of termination of transcription.
I think you are mixing up between prokaryotes and eukaryotes.
Yeah, okay. Yeah, I'm asking about the prokaryotes.
So prokaryotes does not have poly-A tail. They have a U-rich sequence, but not poly-A tail.
Okay.
Poly-A tail is a post-transcriptional modification, which is added to an RNA after it is transcribed. Okay, my clear-cut diagnosis is that none of you have actually listened to the lectures.
Sir, I have listened.
Because the quality of the questions have dramatically dropped. So no point carrying out this charade क्योंकि आप लोगों ने पढ़ाई नहीं की हो. So you tell me, should there be another discussion hour on Friday? I plan to record and upload a series of lectures on translation this week only. Okay. Bolo? Should there be a class on Friday or not? Because I missed Monday's class. Or in other words, will you study more and participate in a more productive discussion on Friday? आप लोगों को अगर पढ़ाई नहीं करना है तो कोई फर्क नहीं पड़ता है. मैं क्वेश्चन पेपर थोड़ा टफ कर दूंगा बस. That's all.
No, so please take the class. Like anyway, if you are not taking the class, we are not reading that much. I don't know, matlab.
It is clear as daylight. हाँ बोलो.
Sir, actually I was looking for the slide. So in the portal of IITK where the lectures are uploaded, I cannot download the slide of these new lectures. So where should I look for them?
Well, there is also a YouTube channel, and also I do not know why. Like I have not changed anything in YouTube.
Oh, actually, I don't know, sir. For me or like others also, first lecture is only visible there, and the second and third lecture is not there. So I watched from YouTube, but I did not find the link at YouTube. The CRISPR-Cas link is attached with the lecture 22, I guess, but I did not find that one.
What?
If you have subscribed Sir's channel,
Yes.
then as Sir uploads a lecture, it
Will
will
get-
come. Like
Okay.
today, Sir has uploaded the last lesson.
You
like
will also get
23rd lecture.
notification.
Yeah, you will get the notification.
Okay, sir. Actually, I watched from the portal itself, that IITK portal. So yeah, I am not familiar with the YouTube channel much, so I will search in it thoroughly.
Yeah, like portal में क्या problem हुआ? I do not know what
Oh,
problem.
actually, sir, check that the last lecture. I don't know if others have checked it. If I am going to see it, I am clicking it.
Yes,
It's
yes.
saying that the video is not available.
Yeah, in mine also,
so
like,
there's lecture
Yes.
21, there's no lecture 22, and when we click on 23, it says that there's no video in this.
yeah, same is the case with me also.
Yeah, I will check. So Piyusha, here is the
Sir,
YouTube
there is also PPT.
channel right now, so
Okay,
that
sir.
opens in a tab.
Yeah.
हाँ. अब
Yes,
बाद में आप bookmark कर लेना.
Okay, sir. Okay, sir. I will subscribe.
Let me quickly check the Mookit thing. See, I do not really use Mookit. I use it only to communicate the marks and for nothing else.
Sir, also can we get the PDF of this slide, these lectures?
It is there, like in the Mukit thing only it is there.
No, sir, it is
No,
not.
No, sir, it was not there.
Sir, only this one is not there. Others are
Others
there.
are there. Only these new sets of lecture, these slides are not there.
Okay. See, I can see 21 and 23, but you are right, not 22. Can you see, can someone check in parallel?
Yes, sir.
I can see 21 and 23, but I cannot see 22.
Sir,
Yes, sir. 22 is not there.
sir, please click the 23 link. It's showing that there is no video lecture for this topic. It is like not opening.
I do not know why. I have no clue. Okay, so let me try this while I am with you. Okay. I just published lecture 22. It was a mistake. I forgot to publish it. Can you see if it is visible now?
Yes, sir, it is visible now.
And is it working?
Yes, sir. It is accessible.
You know, it is, okay, now let me see, lecture 23. हाँ, lecture 23 कुछ दिखा नहीं रहा है, ठीक है. Let me delete it and add it again. That is lecture 23, right?
Yes, sir.
Okay. So in the third lecture on post-transcriptional, just hold on. Start with transport of RNA. So what we have learned so far is capping and polyadenylation of the RNA, splicing of the RNA, and now we are talking about the spliced matured RNA coming to the cytosol. A good way of looking into it is through I was doing something, it did not let me finish. No. Okay, can you check now for lecture 23?
Yes, sir, it's there. Yeah,
Yes,
it's
sir.
playing.
I have uploaded the PPT as well there.
Yes, sir, that also is visible. Yes, sir, got it.
Okay. So Friday 8 p.m.
Yes.
Yes, sir.
Yes, sir.
Shreya, so looks like she's so sad.
Sir, actually,
Huh?
no, no, sir, actually, there is some problem with my audio. That's why it came very low.
हाँ मूवी का
No, no, sir.
हाँ अच्छा ठीक है चलो हाँ
Sir, I just found a few papers regarding RNA I motif, and it forms with strands which are rich in cytosine residues.
हाँ तो वो थोड़ा समझा देना सबको एक दिन हाँ
Yes.
because I do not know how C will interact with C itself.
So actually, it requires two strands, not just one. And there has to be a hemiprotonation of one of the C, that is cytosine residues,
Okay,
for it to interact.
ठीक है
The this problem, like why when we come from the lower organisms to the higher organisms, why we have more number of introns, or why introns crept into the genome of the higher organism? Is there any explanation to this by the research community till now?
Yeah, like there are some. One is that it gives you more robustness against mutation. Hmm. Because majority of the places, if there is a mutation, it does not affect us. While in bacteria, if there is a mutation, it can seriously affect it.
Okay.
Then introns, many genes are encoded within the introns. Okay.
Okay.
So it serves certain higher regulatory functions. But I do not think much more is known about it.
So that may be a reason, like you were showing that graph of the exons are just 100 to 300 kilobase per stretch, and the introns are highly variable.
Yeah.
So this may be a reason of like introns being more like prominent in the genome, like it is less prone to mutations and
That is a possibility, but
possibility.
I would not know. I would not know. I do
Yeah,
not want
like
to comment.
nothing is known clearly about it. Yes.
At least, see, nothing is known clearly is one thing. Nothing is known to me is much more better definition. Nowadays, a few thousands of papers are published in a week. Okay.
Yeah.
And it
Yes,
is
sir.
not humanly possible for anybody to
Possible.
know all of those until and unless it is a big story or it makes it to the newspapers or it comes to the textbook. It will not be known. There will be some obscure publications which can still be very important but obscure nonetheless. And as the good example is your CRISPR-Cas. Like nobody knew of the original CRISPR-Cas papers outside of the field till it became a big deal.
Big deal. Yes, sir.
Right.
Sir, and you also talked about this. You were talking about the experiment of xenopus and HeLa cells. So there you talked about like RNA binding proteins shuffle in and out of the cell.
Now,
So
out of the
where
nucleus.
out of the nucleus, yes, in and out of the nucleus.
Hmm.
So, sir, how that is important in the process of transcription?
Not transcription for RNA export. Like
RNA
all
export.
these
Okay.
RNA exporters like NXF1, NXT1, PCL3, they all comes out of the nucleus, right?
Yes.
And
With the
then they
cyto.
go back in.
Yeah.
So
Yes.
that's precisely what it is.
Okay.
Padhai karo, there is a problem embedded within the PPT. Try to solve it. And I can actually design many such evil things with splicing and RNA degradation, etc. ठीक है ना? चलो पढ़ाई करो. You have not studied today. None of you have studied today. सब वो प्रेजेंटेशन आधा घंटा पहले भी नहीं देखे होंगे. चलो टाटा, बाय बाय.